Assalamu alaikum. Welcome to the beautiful hematopoietic-lymphatic system module. I'll be giving you 7 or 8 lectures related to biochemistry. So this is your host, Dr. Mamoun Ahram. So let's start. In the first lecture, what we will do is talk about hemoglobin. So I'll go over what we learned 2 years ago in the general biochemistry course, and I'll give you more information as well so that you don't get bored. Okay, so let's start by talking about hemoproteins. If you remember, we talked about proteins having non-protein groups attached to them. And these proteins are known as holoproteins. If we remove this non-protein group, then it is known as an apoprotein. Hemoproteins are holoproteins because they have a non-protein group, which is a heme, attached to them. Okay? And hemoproteins, actually, they belong to a large class of proteins with different functions. Probably the most important or most famous is the myoglobin and hemoglobin, which are involved in binding oxygen for the sake of storing oxygen or transferring oxygen. We have cytochrome P450 proteins in the liver that are very important in detoxification of xeno molecules. We have also heme groups being involved in the electron transport chain, and we have other proteins that function as sensors, and heme plays a major role. So what is heme? So heme is an organic molecule. It's composed of 4 rings, and these 4 rings, which are designated as A, B, C, and D, these rings are pyrrole rings. This is the structure of a pyrrole ring. Okay, now, so it is a ring molecule. It's a cyclic molecule overall, and it has branches that extend out of the heme molecule. Primarily, we have methyl groups, vinyl groups, and we have the charged propionate group as well. But overall, other than this group here, the propionate, overall the molecule is hydrophobic. Now, in the center of the heme molecule, we have an iron atom bound to it, and this iron has 6 coordinates, meaning that it can form 6 covalent bonds. 4 of them are with the heme molecule itself. And then we'll talk about the other 2 in a few minutes. Now this heme, the active form of heme has this iron attached to it in the center. If we remove this heme, the molecule, the precursor of heme, is known as protoporphyrin 9. So let's talk about hemoglobin right now. So hemoglobin is a globular protein, and it is actually a heterotetramer, meaning that it is composed of 4 chains, and these chains are different. We have 2 chains that are known as beta chains, and we have 2 chains that are known as alpha chains. So it is an alpha 2 beta 2 protein. Attached to each chain, we have a heme molecule. So we have 4 heme molecules per 1 hemoglobin molecule. Now, just like any other globular protein, we have the hydrophilic and charged amino acids found on the surface, and we have the hydrophobic nonpolar amino acids hidden inside the protein. An exception to the distribution of amino acids, with hydrophilic amino acids to the outside and hydrophobic amino acids to the inside, is 2 histidine residues. which are located right in the core of the hemoglobin molecule. Now, these two histidine molecules play a very important role in the function of hemoglobin. So one of them is known as the proximal histidine because it's closer, and the other one is known as the distal histidine because it's a bit farther. Now, the proximal histidine is covalently linked to the iron atom, so that's the fifth coordinate. And the distal histidine lies on the other side of the heme molecule. Now, and it plays a role in regulating the entry of oxygen when it goes into the hemoglobin molecule and binds to iron. So, something about the heme molecule that I should have mentioned is that the way it's located, remember how I talked about the structure of heme having a propionate group on one end or one side of the molecule. Well, the way heme is located is that it's located inside in the hydrophobic pocket, but the propionate groups extend to the outside, so it does interact with the hydrophilic molecules on the outside on the surface of the hemoglobin molecule. Okay, so we have 4 chains, right? 2 alpha chains and 2 beta chains. Now, the alpha chain is composed of 141 amino acids, whereas the beta chain is composed of 146 amino acids. The last amino acid at the carboxyl terminus of the beta polypeptide is a histidine, so that's a third one, okay? And we'll talk about this histidine. So what happens here is that oxygen comes in, it binds to the iron, okay? And the structure of hemoglobin molecule changes. When one oxygen molecule binds to one heme, to one polypeptide, the structure of this polypeptide changes, facilitating the binding of a second oxygen molecule to the second heme, and that would facilitate binding of a third one even further, and the fourth one, and so on. So this is known as positive cooperativity. That is, binding of one oxygen would make it easier for a second one to bind, and so on. And in the next lecture, we'll talk about the effectors, the regulators of hemoglobin function. Now, if we look at the structure further, it's actually a dimer of dimers, and I made up this term. Okay, so basically you have 2 dimers that are bound to each other. One dimer is known as the alpha beta, and the other dimer is the other alpha chain and beta chain. So it's not like 2 betas and 2 alphas, rather it's alpha beta that is non-covalently linked to the other alpha beta. And this interaction between the two is actually facilitated, the interaction between the alpha and the beta is actually facilitated by hydrophobic interactions, whereas the interaction of one dimer to the other dimer is actually mediated by electrostatic interactions as well as hydrogen bonds, and this is a very important point. Now, so what happens again is that it has been found that hemoglobin has 2 structures. A structure known as the T form, and the other one is known as the R form. These 2 structures, not only that they differ in the orientation or the 3-dimensional position of amino acids, this is due to movement of the polypeptides as a result of oxygen binding, but these 2 structures are also characterized by oxygen affinity, different binding affinity to oxygen. With the T form having a low affinity towards oxygen and the R form having a high affinity towards oxygen. So if you look, this is known as the oxygen saturation curve. So right here in the x-axis, you see the different pressures or different amounts of oxygen versus the saturation of oxygen in hemoglobin molecule. And you can see that as the oxygen level increases, of course, there would be more oxygen bound to hemoglobin. But the point is that this binding curve or the saturation curve looks sigmoidal, which is a characteristic of allostatic proteins, just like hemoglobin, meaning proteins that have more than one shape, okay, which is again the R structure and the T structure. So what is the significance of having a sigmoidal curve right here? It means that there are two structures, and these two structures differ in the T form having a low affinity towards oxygen and the R form having a high affinity towards oxygen. Okay, so these are the two structures again. Now, so why is it that hemoglobin have these two structures? Well, what happens is that you have gradual changes in the quaternary structure of hemoglobin. Okay, starting with a little change in the structure of the heme molecule itself. So remember, proteins have 4 levels of structures: the primary, secondary, tertiary, and quaternary. Quaternary is the overall structure of proteins that are made of more than one polypeptide. Tertiary is the 3-dimensional structure of a polypeptide. And the secondary is the structure of secondary structures like alpha helices, which are enriched in the hemoglobin molecule. And the primary structure is the sequence of amino acids that make up this polypeptide, or the order of amino acids. So the idea here is that we have the heme molecule. And this heme molecule in the hemoglobin molecule is actually bent. The reason is that it's a hydrophobic structure, and there is this repulsion between the distal histidine that is located upward right here and the hydrophobic heme molecule. So the heme molecule is bent. Okay, it has a dome-like structure. So what happens here is that when oxygen comes in and it binds to iron right here in the middle, now this repulsion between the distal histidine and heme is gone, or it's reduced. So the heme takes this flat planar shape. The normal shape of a heme molecule is that it's flat or planar. Now, when the structure of the heme molecule changes, what happens is that it pulls the proximal histidine with it. So it goes upward, as you can see in here. Now, this is a little movement actually in the position of the proximal histidine. It's about 0.4 angstrom. Okay, so this is really little movement. But what happens is that this movement affects the structure of the helix that this histidine, that this proximal histidine is part of. So the alpha helix, the structure or the position of the alpha helix also changes. This changes the tertiary structure of the polypeptide that is now bound to oxygen, and that causes breakage of the electrostatic interactions between the chains, between the dimers. Okay, and this breakage of the electrostatic interactions, it is what converts the T hemoglobin to an R hemoglobin, low affinity hemoglobin to a high affinity hemoglobin. Okay, so again, you have these electrostatic interactions between the two dimers. when oxygen comes in and when it binds, the number of electrostatic interactions is reduced, and that results in the relaxation of the hemoglobin molecule. So it becomes relaxed, and R stands for relaxed. Okay. Now, well, let's zoom in. Let's look at really what happens at the level of amino acids. Okay, so I hope that you will appreciate the beauty of this information. So remember I said that the beta chain is composed of 146 amino acids, and the very last one is known as a histidine. So it's known as histidine 146 or histidine HC3, meaning that it's present in helix H at the carboxy terminus, and it is histidine number 3. Okay, so what happens is that this histidine, the very last one in this alpha helix right here, can form interactions, non-covalent interactions. And these non-covalent interactions are between histidine with the positively charged R group of histidine with an aspartate within the same beta chain. And the carboxy terminus, the carboxyl group of histidine, since it's the last one in the polypeptide, can form the negative charges of the carboxyl group, can form electrostatic interactions with a lysine on an alpha chain. So there are two interactions now that this histidine can form, one with an aspartate and the other one with a lysine on the other chain. So what happens is that when you have this change in the structure of heme that changes the position of the proximal histidine, it changes the structure of the alpha helix. This little movement causes a sliding of the beta chain and the alpha chain. Remember, there's a 15 degrees movement or sliding of these polypeptides. So what happens is that these two interactions are broken. So there's no more interaction between histidine with lysine and aspartate. Rather, what happens is that you have this hydrogen bond that is now formed between aspartate on the alpha chain with asparagine on the beta chain. And this hydrogen bond actually stabilizes the R structure. Okay? So again, this is the histidine, the carboxy histidine right here. You have this interaction with aspartate within the same polypeptide, and you have interaction between the carboxyl group with lysine on the alpha chain. So having these two structures for hemoglobin makes huge significance in the function of this hemoglobin molecule, which is important. It's important for hemoglobin to actually bind to oxygen with high affinity in lungs and release this oxygen so it would have low affinity in tissues. So this is just an illustration for the level of oxygen in different conditions or different circumstances. So in inspired air, we have, of course, there's a lot of oxygen relative to carbon dioxide. Now in alveoli of lungs, it's the level of oxygen is still high. It's about 104, whatever. CO2 is about 40 torr. Now, as blood leaves lungs, the level of oxygen is 100 torr. So this is what when we say that hemoglobin is saturated with oxygen. Now, as the blood or hemoglobin, once it reaches tissues, now in tissues there is very low level of oxygen and high level of still high level of CO2 as a result of metabolism. Now, this causes a shift in the structure of hemoglobin from the R state in lungs to the T state in tissues. So now the interaction between hemoglobin to oxygen is low and oxygen is released. So when blood leaves tissues, the level of oxygen is about 40 torr, the level of CO2 is about 45, and it goes back to lungs and so on. Okay, so now, so this is the saturation curve again. We have this low affinity. At low affinity state, the interaction is low between hemoglobin and oxygen. Not much hemoglobin is saturated with oxygen, so it's about at about so during exercise about 40% of hemoglobin is saturated. Now, whereas in lungs, the when level of oxygen or pressure of oxygen is about 100 torr, most hemoglobin molecules are saturated with oxygen. Okay, now this is really important as well in different altitudes, and we'll talk about this in next lecture, and how it's regulated. The reason is that at sea level we have a lot of oxygen, right? But as you go up to, let's say, to high altitudes, to mountains, for example, the atmospheric pressure is greatly reduced. This results in reduction of arterial oxygen. So instead of 100, it's about 60. So there's 40% reduction in the amount of oxygen in arteries, or the amount of oxygen that leaves lungs. Similarly, the level of oxygen, venous oxygen, is low as well. Okay, so as you go higher, the saturation of hemoglobin with oxygen gets lower and lower, and that causes a person to have hard time breathing. And actually, I was in this area right here about, I don't know, 25-30 years ago, and I was really having a hard time breathing. So I was attending a conference. I wasn't having fun skiing or whatever. Okay. So we talked about positive cooperativity, right? So that's a hemoglobin molecule that is free of oxygen. When one oxygen binds, it makes it easier for a second one to bind, and so on. So the affinity increases as more oxygen molecules bind. And this is the beauty of the hemoglobin molecule having two structures, a low affinity state and a high affinity state, so that in lungs it becomes saturated easily, and in tissues affinity is reduced, and oxygen is released. So the thing is, or the question is, how does hemoglobin change structures? That is, in other words, does it change from T to R, or rapidly or directly, or does it undergo gradual changes? That is, you have T on one end and you have R on the other end, and in between there are intermediate structures. Okay, so these are the two models that tried to explain how hemoglobin changes structures. So one model is known as the concerted model. Concerted meaning that it's rigid, that is either T or R, and that's it. So this model, which appears to be more accurate than the other model that I'll talk about in a second, is that it appears to be more accurate. That is, what it says is that hemoglobin exists in two forms. We have a T form, that is the low affinity state, and we have the R form or the relaxed form. Now T stands for taut or tense. Okay, so and the R form is the one with the high affinity state, right? So what happens is that it says the following. That is, these two structures exist in equilibrium. So T would change to R and R to change. In the absence of oxygen, hemoglobin exists more in the T state than the R state. Now note the direction of the arrows. Okay, so yes, in the absence of oxygen, hemoglobin can exist in the R form, but it's rare compared to the T form. Now what happens is that when an oxygen is added, when it binds to one polypeptide, the equilibrium changes a little bit. So now you would have more proportions of hemoglobin in the R form compared to when no oxygen molecule existed. Okay, now let's add another oxygen molecule. Now notice the arrows again. So you have the equilibrium shifts a little bit also towards the formation of hemoglobin in the R form. You add another one, the equilibrium changes, and when you add the fourth, when a fourth oxygen molecule binds to hemoglobin, now you have higher proportion of hemoglobin in the R form versus the T form, or if you compare this to when oxygen does not exist. Okay, so that explains the positive cooperativity in terms of oxygen binding to hemoglobin. Okay, now there are other effector molecules that can bind at other sites, and they can also shift the equilibrium, and we'll talk about these effectors in the next lecture. The other model says that no, it's not only R and T, there are actually structures in between. So what it says is that when an oxygen, so it says that hemoglobin exists in the T form in the absence of oxygen. When oxygen molecule binds to one polypeptide, it changes this polypeptide to the R form, and it changes the structures of the neighboring polypeptides into something that is in between T and R. When a second oxygen binds, now you have more change in the structure of neighboring polypeptides, and the polypeptide that is bound to oxygen now becomes in the R form, and so on. So when you have a third one, now you have more changes that take place, so it becomes easier for this polypeptide to bind to a fourth oxygen molecule. So these changes, these gradual changes, would also affect the affinity of hemoglobin towards oxygen. So this polypeptide right here, for example, would have very low affinity compared to this polypeptide, and so on. So this model system is known as the sequential or induced fit, or KNF, the abbreviations for the initials of the names of scientists who came up with this model system. So it appears that this model system is less accurate, but it can be true to other allostatic proteins. Okay. So let's talk about the different hemoglobin molecules now, because there isn't only one hemoglobin molecule. There are more forms of hemoglobin molecules. And these forms appear during development. Okay, so what happens is that in the adult stage, you have hemoglobin molecule predominantly made of 2 alpha chains and 2 beta chains. But what we have in here is that actually during development, the hemoglobin molecules change polypeptides. So in the embryonic stage, we have hemoglobin made of two different polypeptides compared to the adult stage or even the fetal stage. So in the fetal stage, we have more hemoglobin made of an alpha chain, two alpha chains, and another chain known as a gamma chain. Now in the adult stage, we have an increase in the expression of the beta polypeptide and a decrease in the expression of the gamma polypeptide. And we have another polypeptide known as the delta, which is not expressed highly in adults. So, and the expression of these polypeptides is actually different during developmental stages. So during the embryonic stage, hemoglobin synthesis starts from the yolk sac, and it is made of two chains of zeta and two chains of epsilon. And this is known as HBE, where E stands for embryonic, HBE goer 1. Because in the later stages, or in the later days of the embryonic stage, you have now expression of the alpha as well, and you have little expression of gamma and some expression of beta. So you have different forms of hemoglobin as well. So you have HBE goer 2, which is made of alpha 2, epsilon 2. You have HBE Portland 1, which is made of zeta 2, gamma 2, and you have HBE Portland 2, that is zeta 2, beta 2. So there are different forms, but mainly it's the HBE goer form that predominates in the embryonic stage. Now, in the fetal stage, what happens is that you have decrease in the expression of zeta and gamma, and you have increase in the expression of alpha and gamma. So the fetal hemoglobin, also known as HbF, is composed of two alpha polypeptides and two gamma polypeptides. Now, if you look at the graph that I showed you earlier, the alpha polypeptide remains to be active, or the alpha polypeptide predominates throughout life. So right before birth, now what happens actually, even throughout the fetal stage, you have an increase in the expression of the beta polypeptide and you have a decrease in the expression of the gamma polypeptide. So at birth, you have 60% of hemoglobin molecule being HbF, that is fetal hemoglobin, and 40% is the HbA, or the adult hemoglobin molecule. Now, but then it changes as we grow, and the fetal hemoglobin becomes only 1% in adults. Now, so the adult hemoglobin is known as HbA. And there are two forms, by the way. We have HbA1, which is mainly it's a tetramer of two alpha chains and two beta chains. That's the predominant hemoglobin. And we have HbA2. And this hemoglobin is composed of two alpha chains and two delta chains. But it seems that the promoter region of the delta gene is not very active. Now, what's important about HbA1 specifically is that it can be glycosylated, specifically at a valine. And this glycosylation is important. So you have a glycosylated hemoglobin, and it's known as HbAc, or specifically HbA1c. Now, this glycosylation of hemoglobin is important because it can be used as a marker for diabetes mellitus. So diabetic patients would have higher proportion of glycosylated hemoglobin versus normal individuals. So the level of hemoglobin, glycosylated hemoglobin, can be used as a marker for diabetic patients. So usually, in order to, there are two tests for diabetes. The first one is the blood fasting glucose level, where basically a person would fast overnight, next day a test is made, the blood is drawn, and test for total glucose level is measured. And it should be between 80 and 120. Now the thing is, the advantage of this test is that it tells us exactly at the very moment of the test what the concentration of glucose is. So if a person eats, for example, eats breakfast and goes to check for glucose level, it would be high, of course, and that's why a person should be fasting when this test is done. Now, looking at the amount of glycosylated hemoglobin is important. So doing the HbA1c test is important because it tells us about the long-term trend. Now glycosylation of hemoglobin does not occur overnight. Rather, it occurs within like a span of time, within a period of time, about 3 months or so. So the thing is, it tells us the level of glycosylated hemoglobin. It tells us more information about the status of this person within the last 3 months. So if a person is diabetic, this person would have high level of glycosylated hemoglobin. If a medication is given to that person, and to check if the medication is working or not, and to check actually if the patient is adherent to the medication, we look for the level of glycosylated hemoglobin. Now, the amount of glycosylated hemoglobin can be expressed as percentages, but the new value or the new test use what is known as the IFCC unit. Now, of course, there are limitations to the glycosylated hemoglobin test, and that is it doesn't really capture short-term variations. It doesn't tell if a person is hypoglycemic or hyperglycemic at the time of the test. Okay? So this table is for you to memorize actually. So normal individuals would have levels of glucose between 80 and 120. So that's normal individuals. Okay. Now this is looking at glycosylated hemoglobin. The DCCT units are percentages. So a person having 5% HbA1c is, or 6%, this person would be normal. What these percentages mean is that 5% of hemoglobin is glycosylated. Now the IFCC units rely more on millimole of hemoglobin molecule per mole of total hemoglobin. Okay, so again, it's a percentage, right? But it's more accurate than the percentages because right here you have 5 and 6. Whereas in here, but you don't have things in between, so you wouldn't really have accurate measure for 5.5 or 5.6. But using the new digits or the new units, I'm sorry, you would have more variation, more indication for the actual amount of the glycosylated hemoglobin. So a pre-diabetic person would have high level of glucose. This is fasting glucose. But if you look at the percentage of glycosylated hemoglobin, so at 7% and 53 millimole per mole of total hemoglobin. Diabetic person would have 8 and higher, okay? And really severe diabetes would have very high level of glycosylated hemoglobin as well as glucose, fasting glucose level. This is severe cases, okay? High risk individuals. So in this section, we'll talk about the genetics of globin synthesis. This switch or transition from the zeta and epsilon to the alpha and gamma to the beta and delta and so on. So it's all about genetics. Okay, so we'll talk about the genetics of globin synthesis, right? So these genes actually exist in clusters on two different chromosomes, and we have two clusters. We have the alpha gene cluster, and it contains two genes of alpha. So you have alpha 1 and alpha 2 right here. You have alpha 1 and alpha 2, and you have a zeta gene. So this cluster right here exists on chromosome 16. And we have the beta gene cluster that contains the epsilon, and we have two gamma genes, and we have the delta, and we have the beta. And notice that the order of these genes reflects the order of expression. So you have expression of the zeta and epsilon, and then we have a switch to the gamma for the beta gene cluster, and to the alpha in the alpha gene cluster, and then to the delta and beta. Now note that these are located on two different chromosomes as well. So you have this genetic switching that takes place, okay, from zeta to alpha, and it's all timed, and from epsilon to gamma again to delta and beta. And this is determined really by genetic factors and transcription factors, and it's all timed. It's all controlled. Now, what's interesting as well is that premature newborns follow their gestational age. That is, if you have this switch that takes place at birth, well, at the 9 months, same thing with premature babies. They follow the same exact timing as well, which is quite interesting. So how is switching done? This switching of transcription or expression of these genes from one to the other. Well, it's all controlled by a number of activators and silencers or repressors, okay, at regulatory sequences. Okay, so what happens is that in the alpha gene cluster, it contains a region, an enhancer region known as the HS40, and it lies upstream. If you remember what this term means, it lies upstream of the alpha gene cluster. So with DNA looping, you have proteins that bind to this enhancer, and these proteins can interact with regulatory elements that are present at the zeta gene, and then they can bind different co-regulators would bind to regulatory proteins on the promoter region of the alpha gene, and so on. So you have different expression of these regulatory proteins that take place at different stages of development. Now for the beta gene cluster, there is another regulatory region or enhancer known as the locus control region, and this region also lies upstream of the beta gene cluster. And again, it's the same idea. You have proteins that bind to this region, and then DNA loops around and can interact with proteins that bind to the promoter region or the regulatory sequences that lie ahead or upstream of the different genes. So in the fetal stage, you have interaction between proteins on the LCR region with regulators or regulatory proteins that are bound or control expression of the gamma genes. And then as you see the switching from the fetal stage to the adult stage, you have the interaction between these proteins that exist on the LCR region with other proteins that bind to regulatory elements of the beta gene and so on. Okay, and it's all timed again, and this timing starts with fertilization. So what happens here is that you have modification of these of the DNA regions by epigenetics. Remember, acetylation, methylation, and so on, as well as chromatin compactness or packaging. Okay, having euchromatin versus heterochromatin, and so on. So the idea is that think about it in terms of treatment. Right? So people with defects in the beta gene will, like in thalassemia, for example, beta thalassemia, what if we treat them by inducing expression of the gamma gene, producing gamma polypeptide? That would solve a problem. Now, something else that I should have mentioned as well is that note that in the alpha gene cluster, by the way, we have 2 genes. Each chromosome contains 2 genes of alpha, whereas with the beta genes cluster, we have 2 beta genes.